Hi everyone. I'm BTech Biotechnology student of CBIT. In this video, I'm going to talk about my final year project of using fluorescence spectroscopy to evaluate the tertiary structure of maps. This work was carried out at Dr. Reddy's laboratories. This project was aimed to evaluate the native tertiary structure of map and study the thermal stress effect on its tertiary structure. Proteins are three-dimensional arrangement of amino acid chains. The primary structure of a protein, its amino acid sequence, drives the folding and intramolecular bonding of the linear amino acid chain, which ultimately determines the protein's unique three-dimensional shape. Antibody, also known as an immunoglobulin, is a large Y-shaped protein produced mainly by plasma cells that is used by the immune system to neutralize pathogens such as bacteria and viruses. MAPs are antibodies that are made by identical immune cells that are all clones of a unique parent cell. Therapeutic proteins are produced in a live cell as macromolecules which are purified and formulated. Heterogeneities of these proteins always exists while producing them. Hence, the evaluation of protein folding and unfolding is of high importance as they affect the affinity of the drug. Aromatic amino acids phenylalanine, tyrosine and tryptophan have naturally fluorescence property which can be used to study their tertiary structure. Phenylalanine and tyrosine emits fluorescence relatively weakly compared to tryptophan. Emission of both tyrosine and tryptophan residues is observed when protein fluorescence is excited near the absorbance maximum around 280 nanometers. Excitation at or above 295 nanometers gives emission spectrum of tryptophan almost exclusively. Tryptophan residues are particularly valuable intrinsic probes since the indole ring is very sensitive to its environment and since there are often only a few tryptophan residues in a protein. Fluorescence activity can be explained with this classical Jablonski diagram where the molecule is first excited by absorbing a photon from its ground electronic state to one of the various vibrational states in the excited electronic state. Collisions with other molecules cause the excited molecule to lose vibrational energy until it reaches the lowest vibrational state from the excited electronic state. As some energy is always lost by non-radiative processes, the energy of the emitted light is always less than that of the absorbed light. Hence, the fluorescence emission invariably occurs at a longer, less energetic wavelength than absorption. Two monoclonal antibodies belonging to same superfamily and one fusion protein were used in this study. The sample of high concentration is initially diluted to an intermediate concentration 1 mg per milliliter and then were diluted to the final concentration of 0.5 mg per milliliter for steady-state fluorescence measurements. The concentration was measured using UV visible spectrophotometer. Fan fluorescence emission spectra were collected using an excitation wavelength of 295 nanometers to avoid excitation of tyrosine. Emission spectrum was collected in range 300 to 440 nanometers. The emission profile was plotted using excitation and emission intensities. At 130 nanometers, tryptophan is in a properly folded form while at 350 nanometers, tryptophan is exposed to solvent. Hence, monitoring this ratio helped to observe conformational changes. The fluorescence emission of tryptophan of maps excited at 295 nanometers when plotted gave a profile as seen in this graph. The emission increased with wavelength and after reaching a maximum it started decreasing. A fusion protein is a chimeric product of a fusion gene. It is different from MAPs which can be seen by comparing emission profiles of MAP and fusion protein where the intensity of fusion protein is relatively less than MAP as seen in this graph. After thermally stressing the samples were 0, 6 and 15 days at 40 degrees Celsius, the tertiary structure was evaluated. It was observed that the emission maxima were not altered even after thermally stressing the samples for 6 and 15 days. It can be concluded that although all the three therapeutic proteins are similar, the overall three-dimensional structure is very different. The variation in tryptophan emission maximum can be clearly seen. The thermal stress study revealed that the tertiary structure of MAB was not perturbed even after 15 days. Drug product development of proteins is a challenge for pharmaceutical teams. Dealing with biopharmaceuticals in specific due to the different nature of drugs encountered is very difficult. The study demonstrates the valuable information that fluorescence spectroscopy, as an analytical tool, can provide about monoclonal antibody tertiary structure, which helps in further development process. The thermal stress study also provides valuable information on how temperatures during storage can impact the tertiary structure, which in turn affects the affinity of drug. Thanks for watching.